Thank you for that introduction. It's a pleasure to be part of this meeting. In the Office of Biotechnology Products, or OBP, we're focused on supporting quality aspects of drugs, specifically biotechnology products. Our goal is to ensure release of consistently safe and potent protein products and to uphold standards of purity. I'll begin by stating that this presentation reflects our experiences as reviewers and should not be used in place of regulations, published FDA guidance, or discussions with the agency. For the first part of this talk, I will focus on review principles for biotechnology products, and then my colleague, Dr. Kristen Nickens, will discuss communications with the agency. We're beginning with the statement, know your product, with an emphasis on your. And that's because one of the themes here is that all of these products are unique with their own considerations. Appropriate control relies on an overall strategy guided by product understanding. For reference, I've listed here the current laws governing regulation of biotechnology products. To give you a flavor of what we see in OBP, our product portfolio includes a wide range of antibody-based products, from commonly used classic immunoglobulins and fragments to many novel monospecific and bispecific constructs, including T-cell engagers. And then, of course, we also see a wide variety of non-antibody-based products. These include enzymes, cytokines, growth factors, soluble receptors, toxins, and now also products that under the Affordable Care Act have been designated as protein products and regulated under BLA, which include insulins and the smaller growth factors. These proteins can be modified in various ways, such as pegylation to enhance half-life, conjugation with radioisotopes for imaging or radiotherapy, or, for example, also with drugs. And we continue to see an increasing number of antibody-drug conjugates under study in oncology. There are a number of challenges inherent to protein products. First off is the sheer size. And to bring this home, this image represents one of two FAB portions, or antigen binding regions, of a classic antibody structure, so approximately 1/3 of the full antibody. To give you an idea of the relative size, it's shown here in comparison with statin. The estimated range of our products is about 4,000 to 300,000 Daltons. Within these molecules, we're dealing with multiple levels of protein structure and multiple active sites that require the proper conformation for full functionality. This often involves post-translational modifications and can yield a heterogeneous mix within a single vial of the product. The goal is controlled heterogeneity and to maintain those features critical to the activity of the molecule. In addition, protein products are susceptible to a variety of degradative pathways, and it's important to take steps to ensure product stability. Control of product quality is largely predicated on understanding the relationship between structure and function. This involves extensive physicochemical characterization, bioactivity assays, which are usually cell-based and which ideally reflect the product's mechanism of action, control strategies with defined release specifications that capture the critical attributes of the molecule, and control over the manufacturing process. In addition, manufacturing changes are common, and it's necessary to link the pre- and post-change product to establish their relationship. I've broken these down into three main areas, starting with product characterization. There are many established methods that can be used for product characterization. I've listed some of the approaches here as examples of methods that can be used to measure size and charge heterogeneity, post-translational modifications such as glycosylation, as well as bioactivity. When we review your submission, we look at your assays in the context of your overall strategy. We expect you to select the most appropriate assays to meet your needs and to justify the selection. Lots should be compared to an established reference material. For most of our products which don't have established standards, the reference material is often initially derived from an early toxicology lot. For products with endogenous counterparts, for example, replacement therapies, those should also be compared as appropriate. Through the course of product development and while progressing through different phases of clinical trials, an aim of these characterization studies is to accrue knowledge on how product quality attributes influence clinical outcomes, so that ideally by the time of licensure, the range of acceptable values for maintaining clinical efficacy and safety is well defined. I'd also like to point out that as knowledge unfolds with technological advances and increased biological understanding, we may ask for further characterization to address new structure, function, or safety-related attributes as they are recognized. I'd like to take a few minutes to talk about the bioassay. These assays are the only assays that can directly measure the biological activity of biological therapeutics, and they serve as a measure of the product potency. The reported activity is controlled by using a reference material in each run and can be expressed in units or activity relative to the reference material, as is commonly done with monoclonal antibody products. These assays are required for biotechnology products under the Public Health Service Act. Because of the complexity of many of these molecules, it's understood that in some cases it may be the best way, even with extensive physical-chemical characterization, to confirm higher order structure of the product using an assay that tests its activity. Though mimicking the biological activity in the clinical situation with an assay is not always possible, the bioassay is ideally based on the mechanism of action of the product that's believed to contribute to the mechanism of efficacy. A correlation between the expected clinical response and dose and the activity in the biological assay should be established in clinical studies. If there are multiple possible mechanisms, the structural requirements for the different activities may differ, so more than one assay may be required. Similarly, a product targeting more than one indication may utilize distinct mechanisms for each indication. Therefore, the same product may have different bioassays for the different indications. There are a wide range of approaches that can be used. Some examples are shown at the bottom of the slide, and one might consider a number of factors in selecting the assay, including ease of validation and adaptability to high throughput. Bioactivity is a critical parameter, and it's a good idea to identify the bioassay earlier rather than later. Recognize that working with less well-described assays may require more attention early in product development. With regard to relating product quality to safety and efficacy, your product may include a number of product-related variants. This mix of product variants may represent product-related substances, which have the properties comparable to those of the desired product with respect to activity, efficacy, and safety, but also product-related impurities. These arise during manufacturing or storage and are either uncharacterized or do not have properties comparable to the desired product with respect, again, to activity, efficacy, and safety. Examples of product-related impurities would include fragments or incorrectly folded proteins with only partial potency, glycovariants, which alter binding affinity, so can alter activity or pharmacokinetics, and aggregates, which can alter potency and could also be a safety concern by leading to immunogenicity. And you'll want to control these variants by in-process or release testing. At the heart of this is having assays that are capable of recognizing these differences. So you'll want to consider, are your tests capable of detecting modifications that might occur in the product? And are they capable of detecting all impurities that might occur? It's important to establish sensitive and reliable assays as early in development as possible. We also recommend that you retain samples so that if your assays change, you can bridge results across assays. One difference between the regulation of biotechnology products and small molecule drugs is that under the Public Health Service Act, the licensure of biologic products encompasses the manufacturing process and facility, and I will discuss that next. Quality can be impacted by steps across the manufacturing process, starting with your choice of expression construct containing the coding sequence, the species from which the gene is obtained, and the cell substrate used to express the product. There are a variety of cellular expression systems commonly used, including microbial cells, such as bacterial and yeast, and higher eukaryotic cells. Most commonly we see mammalian cell substrates, but also insect and plant cells. These systems vary in features so that you can pick what's best for your product. That might include optimizing for post-translational modifications if relevant, or which system is best at achieving the proper protein folding. You might also consider, among other factors, speed of development and productivity. One of the key concerns with the manufacturing of these products is the potential for adventitious agents due to their production in living cells. Proteins can't be terminally sterilized, so it's critical to maintain control over the process. Be aware that contamination with pathogens can come from the cell substrate, from raw materials which may or may not be animal-derived, or from the environment. Following the harvest of product yield from producer cells, there's a purification process that typically includes 2 to 4 chromatography steps, which can change the product profile, as well as virus reduction steps. Formulation also has a dramatic impact on product quality and stability. And as I mentioned earlier, the manufacturing process and facility are part of the license application. There's typically a pre-licensing inspection during the BLA review cycle, and also prior to approval of post-marketing supplements for a new facility. I've spoken about product-related impurities. Process-related impurities can also be present, and some examples of common process-related impurities are listed on this slide. They may include from cell culture, inducing agents, selectable markers, or antibiotics that were present in the culture media, components of the producer cell lines, such as host cell proteins and DNA. There's the potential for adventitious agent contamination and endotoxin, and also leachables, such as from chromatography resins or the container closure systems. The manufacturing process is designed to remove both product and process-related impurities. From a safety perspective, a critical aspect of this is the sourcing of raw materials to prevent introduction of adventitious agents. We also recommend that for tissue culture medium, when possible, you use chemically defined components. We encourage sponsors early on to establish a 2-tiered cell banking system of a master cell bank for long-term storage that's used to develop a working cell bank from which you manufacture. These cell banks should be qualified to be free of contaminating microbes and adventitious viruses. It is acceptable to start a phase 1 study with material manufactured from a master cell bank and to add a working cell bank later in development. The end of production cells and unprocessed bulk harvest are tested for impurities, and clearance of adventitious viruses must be demonstrated. Testing is performed throughout the manufacturing process to confirm removal of process and product-related impurities. It's important to implement microbial control throughout the process and to ensure adequate cleaning of reusable components or to use disposable systems. These are some examples of the kinds of manufacturing-related issues we've seen. They can happen as a result of any step of the process. For example, if a producer cell line isn't appropriately cloned, you can get selection of cell populations that give rise to new product variances that may have sequence differences. This is the kind of thing that happens early on, but that may not be recognized until later stages of clinical development, which can be problematic. Changes in medium components can alter producer cell growth characteristics or can alter post-translational modifications, which can impact the activity of the product. One can get co-purified proteins that come through the purification process, We've seen product degradation that was traced to the presence of proteases from the producer cell line that co-purified with the product and allowed for breakdown during storage. Finally, as an example, at the drug product stage, aggregation and precipitation are often observed if formulation is not optimal. Continuous monitoring of product quality is achieved by lot release testing for both drug substance and drug product. This maintains acceptable levels of critical features that you've identified and restricts the presence of impurities. It involves establishing acceptance criteria based on your product knowledge and manufacturing capability. Early in development, it's understood you'll likely have limited data, and this can be challenging. But it's important to establish acceptance criteria to maintain consistency of product quality based on the information available to you at the time, also taking into consideration the planned clinical study to ensure that the product used in the clinical trials will give you meaningful clinical results. Generally, acceptance criteria can be broader at phase 1, but as clinical development proceeds, they should be reassessed and refined based on your additional knowledge of the product. However, I should note that there are circumstances, as an example in the case of a product with a narrow therapeutic index, when it's important to implement narrow acceptance criteria even early in phase 1. Acceptance criteria are based on the product characteristics, the manufacturing capability, and the assay capability. This slide shows an example of how product knowledge is applied to establishing acceptance criteria. So on the left, we have an ion exchange chromatogram that shows purity of a monoclonal antibody product by charge variance, acidic species on the left, basic species on the right. In this example, the 3 major peaks have been evaluated for content and also for bioactivity using a cell-based potency assay. Peaks 1 and 2 are shown to have full biological activity, whereas peak 3, a variant in peak 3, was found to have lower potency. In a situation like this, one would control the levels of the less potent variant to maintain product quality. These kinds of controls are further defined and refined as product development continues. Proteins can break down through multiple pathways, some of which are listed here. During characterization, we encourage you to stress your product to identify pathways of degradation and to identify stability indicating methods. These should be incorporated into a stability protocol to monitor protein stability during storage. Finally, I'd like to address comparability assessments. These are often needed during the product life cycle due to implementation of major manufacturing changes, such as changes in critical reagents, scale up in production to increase supply, or introduction of a new facility. Sometimes these are done to improve the process for better control. Sometimes you're not able to plan for the need for these. The change might be due, for example, to the loss of a supplier of critical reagents. Now, it's not feasible to have a clinical trial with every manufacturing change, so we rely heavily on analytical assessment to determine the potential for product impact. This reinforces the need to understand as much as possible the relationship between protein attributes, clinical safety, and efficacy to allow us to predict clinical comparability. Comparability assessments typically involve comparison of lot release test results between pre- and post-change material, as well as additional characterization studies. We generally like to see side-by-side analyses, and co-mix studies can be sensitive at detecting small differences between pre- and post-change material. A comparison of in-process testing can demonstrate the efficiency of discrete manufacturing steps, and trending analyses are important to view quality parameters over time. In addition, changes can be detected by patterns of forced product breakdown. If there is a change, depending on its nature, it may call for additional animal testing or possibly an additional clinical study. This decision relies heavily on product knowledge, and it's a discussion we have with our colleagues in the other disciplines. I'd like to emphasize that the extent of the data required to establish comparability is relative to the stage of clinical development and relies on available knowledge based on clinical experience with the pre-change material. A more comprehensive analysis would be expected during pivotal trials or following licensure, for example, than what would be needed for establishing differences between non-clinical to phase 1 material. Manufacturing changes can result in unanticipated product-related variants for which the clinical impact is unknown. So it's important to know the range of modifications for which there is clinical experience. The more you know about the critical parameters in the manufacturing process, the better you can predict the potential impact of changes on the product. And the more knowledge you have on the critical quality attributes of your product, and the better your assays, the better you can predict the clinical impact of those differences. We do recognize that with the new drug program, you will not know all of this up front, but you'll accrue knowledge as your product proceeds from early non-clinical studies through safety and efficacy trials to licensure and even post-approval changes on how product quality attributes influence clinical outcome. This knowledge is gained through process changes, through scale-ups, through optimizing the process for commercialization, as well as associated development studies that correlate quality attributes to clinical impact. It also draws from the shifting knowledge landscape about particular product classes. At the same time, you'll be able to refine your specifications as you optimize and validate the assays. So in summary, biotechnology products can be heterogeneous mixes. The goal is to ensure safe, pure, and potent product, essentially controlled and consistent heterogeneity. This is achieved by control of the manufacturing process and through product quality release testing. The final message here is to start as early as you can in the development process to understand the critical attributes, establish a testing strategy that captures the most critical features of your molecule relevant to mechanism of action and clinical efficacy to ensure consistency. Each product is different, and each test provides certain information about the quality attributes of a product, but no one single test completely characterizes all aspects of a protein product. It's a matter of establishing a comprehensive overall strategy. So the key message, know your product, control your process. This is my final slide with useful resources related to the CMC of biotechnology products. My colleague, Dr. Kristen Nickens, will now discuss interactions with the agency to facilitate product development. I'll be back for a Q&A with Kristen. Please feel free to enter questions in the chat box.
Thank you, Wendy, and hello. My name is Kristen Nickens, and as Dr. Weinberg conveyed, a key aspect of a successful CMC strategy is knowing your product. With a thorough understanding of your product and process, you will be able to discern product quality nuances related to your process and any changes that may occur. However, with changes comes risk, and with risk comes uncertainty, and with that uncertainty, you may have questions. And so when these questions occur, you're encouraged to meet with the FDA and discuss your development strategy. And so we believe that in addition to knowing your product, that communication is a key aspect to a successful CMC strategy. So within my portion of our discussion, I will be focusing on communication strategies and approaches that may be useful for you as you continue or begin your product development programs. I'll also focus on some common product quality topics that are discussed with the FDA and give you a case study on each one of those. So throughout the talk, the key points that I really want to highlight are: why communicate with the FDA in the first place? When should you be communicating? How should you communicate? And what is the right thing to discuss and communicate with the FDA around your product development process? Why communicate at all with the FDA? Well, you can find that these communications allow you to get regulatory feedback, specifically on CMC strategies that you may have, You'll also be able to identify and discuss potential pitfalls and avoid roadblocks as your development program proceeds and with respect to changes you make. You'll be able to exchange scientific ideas and discuss novel approaches with the agency. And this can also result in a more efficient and robust development program, which is a win-win for us all. A little bit about when the right time is to communicate with the FDA. Well, any time is really a good time during your drug development program. However, we generally recommend that you come to us during critical milestones such as end of phase 1, end of phase 2, pre-BLA meetings. However, if you have major changes or unique control strategies that you plan to implement, these may also be good times to come and discuss these changes with the FDA. Also, you want to consider your development timeline when you decide to reach out. And so it's very important that you kind of implement these communication points within your program, develop your product development program. How should you communicate with the FDA? Well, there are a variety of pathways depending on the type of product you have. For example, for PDUFA products, we have a Type A, Type B, and Type C meetings that depend on the type of questions you have or the phase that you're in. Will depend on what type of meeting you will have. There's also a very similar pathway for biosimilar product development. Also, we have in the Office of Pharmaceutical Quality emerging technology program for products that are unique. And so this particular program is implemented to promote the adoption of innovative approaches to pharmaceutical product design and manufacturing. It's focused on the CMC and facility-related sections of applications. So if you're interested, you can reach out to the CDER ETT email address listed here, and there's also a guidance document which is included at the end of this slide deck for your information purposes. A few types of the technologies that are typically seen in the emerging technology program include continuous manufacturing, process analytical technologies, biosensor development, as well as a variety of novel dosage forms. Very interesting program. So knowing your product should really tell you what to communicate, should really help inform you on what to communicate with the FDA. Taking a risk-based look, a risk-based approach at identifying those issues or those changes that can impact your product's quality are those things that you can hone in on to really address and focus your communications around. Also, understanding where you are in your product development can focus your questions and around what you should be communicating. For example, if you are entering into a later phase of development, then it may be that appropriate time based on the information and data understanding of your product that you have to ask questions that are regarding late stage development. However, that particular approach might not be appropriate if you're in an earlier stage of development. And so that information may not be able to allow the agency to give you targeted and well-rounded phase-specific feedback about your product. So some common product quality related topics that we generally find sponsors requesting meeting discussions around include process development approaches or changes, such as scaling up a process or implementing a variety of changes to their manufacturing processes, product development approaches, such as changes to their formulation or changes to their drug product presentations, such as going from a vial to a prefilled syringe, as well as analytical method changes. And so in the following slides, I'm going to present some case studies that kind of fall into each of these different buckets. I'll begin by presenting the proposed changes that the sponsor is planning to implement. as well as the information and data that they include in their briefing documents that are submitted to the agency prior to that meeting. And then the FDA's response, I'll go over as well. And finally, summarize each case study with some key takeaway points that are related to those particular discussions. So let's start with the process development case study. In this particular case, the sponsor is proposing to implement some process changes to support the initiation of their phase 2 material into clinical studies, and they will also propose to use the material for future commercialization. Some of the changes they're proposing include changes to a new cell, changes to their cell bank, an increase in production scale, a drug substance manufacturing site change, an increase in their drug product fill volume, and changes to their packaging components. Their question is, does the agency agree with the overall comparability approach that they're proposing in their meeting package? And so the information that they provided was a description of the comparability study, which included a brief description of the changes, the number of pre and post change drug substance lots they plan to include in the comparability study, and then the list of tests that they also will evaluate the product quality data, including in-process samples, release tests, extended characterization studies, and stability evaluations. The response from the FDA, based on the information they provided in their briefing document, was that their overarching comparability approach appeared reasonable. However, because they did not provide information on all of the changes, specifically the cell bank, they were not able to provide targeted feedback regarding that particular change. They also were not able to provide feedback on the particular approach to evaluating the acceptability of the comparability study because the sponsor did not describe that in the briefing document. Further, the FDA identified that some of the process changes may have the potential to impact the virus clearance capacity of their process, and recommended that the sponsor evaluate the impact of their process changes on virus safety. And finally, they highlighted that it's premature to comment on the adequacy of these proposed changes to their process and the resulting material to support commercial use, because that information, that level of information and data, were not provided in the meeting package, and it's still very early in their development process. So the takeaways from this case study are to perform a 360-degree risk evaluation of the impact of the changes on product quality, to provide sufficient information and details in your briefing document to enable the FDA to give you comprehensive feedback, and to ask early about major changes so that you can get feedback on areas that you may have missed. So let's move on to the product development case study. In this particular study, I'll go over as this particular sponsor has come in with 2 meetings. So I will focus on meeting 1 and then move on to meeting 2. So under meeting 1, the sponsor described their product, which is a drug product that's an enzyme, and it's formulated as a frozen solution. They have some stability study data that showed that over the course of long-term storage at this frozen temperature, they see a reduction in purity and an increase in aggregation and subvisible particle formation. So now they're going to propose to change the formulation to a refrigerated liquid, and they also would like to include polysorbate. Their question is, does the agency agree with the addition of polysorbate to reduce aggregate formation? The information they provided in their briefing document included data from those long-term stability studies, as well as the development study results that show when they add the polysorbate to the formulation, they seem to get a decrease in that aggregate and particle formation. The FDA's response was, it does appear that the particle formation is a major stability problem, and we agree with that. And it does look like the addition of the polysorbate improves the aggregate formation in of the product. The FDA then goes on to recommend studies for future development to support the implementation of this change, such as evaluating the effect of polysorbate on the product's activity, optimizing the levels of polysorbate in the drug product formulation, evaluating the impact of the polysorbate on interpretation and performance of the analytical methods, and investigating the potential polysorbate-related oxidation of their products, and then implementing scientifically justified specifications for polysorbate in their drug products. So the sponsor took this information back and implemented some of their proposed changes. And then about a year later, they came in for a second meeting where they conveyed that they intended to develop the drug product as a refrigerated liquid formulation with polysorbate, but found that even under these conditions, the aggregation and particle formation persisted. So they planned to develop a lyophilized formulation now. Their question this time was, does the agency agree with this proposed change, and does this impact any of the previous advice that they got from the agency with respect to the addition of the polysorbate? Also, are there any additional considerations that they should be aware of considering this new lyophilized formulation? The FDA response was that there are no oppositions to the proposed development plan of a lyophilized formulation, and that the sponsor should consider providing or performing comparability studies between the pre and post lyophilized material and submit that data to the IND prior to the initiation of clinical studies with the lyophilized drug product, and that the previous advice remained applicable with respect to the evaluation of the potential impact of polysorbate on their product and process control strategies. So the takeaways from this include ask early and do not be afraid to follow up. That the continuous understanding and product characterization allows you to make risk-based product changes and to identify issues and points of concern early. And also to apply previously received feedback from the FDA into your continued development and communication approaches with the agency. So our final case study will be an analytical method case study. In this particular case, the sponsor developed a cell-based ADCC assay, which is a presumed mechanism of action of their product. The assay was specific for target 1 and was intended to be included in their lot release. However, they've been having significant development challenges with respect to the development of this assay. Here in the meeting package, they're proposing to redevelop the assay. Instead of using their initial target 1, they will propose to develop the assay using a second target. And importantly, they state that they do not plan to implement the potency assay that they're developing as a release test prior to the initiation of pivotal studies as previously recommended by the FDA. Their question is, does the agency agree with this approach? In their briefing documents, they included the analytical method development study data and reports showing that they've been having these challenges, a description of the newly proposed assay, and justification to support implementation of the target 2 as a target in their bioassay to support the potency and the mechanism of action of their product. The FDA's response was acknowledgement of the efforts with respect to the analytical method development challenges. However, they continue to recommend that the sponsor control for ADCC activity, and being that it's a primary mechanism of action of their product, and that assay be in place prior to pivotal studies. The FDA then went on to suggest that the company identify alternative approaches to control ADCC for their product until that cell-based assay is available for implementation. For example, exploring links between Fc gamma R binding, glycosylation, and ADCC activity. A key point that the FDA made in this response was the importance of appropriately storing retained samples of clinical samples that are taken over the course of development, so that once the new assay is implemented, they're able to bridge the data generated from the old assay to the new assay, which will ensure batch-to-batch consistency and interpretation of their clinical studies over the course of their development with respect to their potency. Finally, the agency advised the company to request a CMC-only meeting in the future to discuss their proposals and their development plan as data become available with respect to this cell-based assay development. So the takeaways here include roadblocks can and likely will occur over your development program. However, if you have a good understanding of your product and your process, you can identify those roadblocks and discuss those strategies early. For example, in this case, prior to pivotal studies. Also, you'll be able to potentially receive feedback on alternative approaches that you may not have considered prior to the discussion with the FDA around your strategy. And also highlights that CMC-only meetings are possible and can allow you to have targeted and more in-depth specific conversations with the agency around product quality related issues. So the takeaways on the communication tips from this talk are really about knowing your product and how knowing your product can help you target what you should ask to the agency with respect to your particular process and product development strategies. To focus your questions and ensure that they are appropriate based on phase-specific considerations. To provide sufficient information in your briefing documents so that the agency is able to provide you with comprehensive and specific feedback. And to learn from that feedback, implement it into your programs as you see fit, and also follow up if needed. Finally, purposefully utilize the available communication tools. Think about those tools as you begin your product development and implement the points in which you plan to communicate with the FDA at those particular milestones. Here is a list of guidance resources that may be useful with respect to meetings with the agency. And finally, Dr. Weinberg and I would like to acknowledge our colleagues in the Office of Biotechnology Products and Office of Pharmaceutical Quality that have helped us in the preparation of these slides. And finally, we thank you very much for your time, and Dr. Weinberg will now rejoin us for any questions that you may have. Thank you.
Thanks, Dr. Weinberg and Dr. Nickens. Those were cogent, concise presentations, and I really like the whole controlled heterogeneity, no doubt a little bit of controlled chaos, and we address all that with some controlled communication. I thought those were excellent presentations. Kristen, I wanted to focus you back, if we can, and ask a question with regards to, can you provide examples of CMC information or lack thereof that might result in clinical holds for first-in-human trials? What are the things that are missing that's like non-starters?
Hi, Jeff. Yes, thank you for that question. So in general, for first-in-human studies, from CMC perspective for biological products, we're really looking at aspects of the safety of your product. And so because, as my colleague Dr. Weinberg mentioned, a lot of the products are developed in mammalian cell lines, we're looking for control and the lack thereof of adventitious viruses. And so the safety testing that we look for is critical to support the adequacy of your phase 1 material, your first in human material. Some of the things that are generally we see lacking in a phase 1 IND that may result in clinical hold can include the lack of sufficient safety testing of the cell banks that are used to produce the product, as well as safety testing of the unprocessed bulk drug substance material. A large piece of supportive data and information that we use to evaluate the safety of your product are virus clearance studies, which we generally see using some type of scale down model system. And those data are evaluated with respect to your proposed clinical manufacturing process. Additional pieces may be some aspects of your manufacturing process controls, like specifications. We would definitely expect controls over potency and purity, as well as identity. And then, as some of my other colleagues have mentioned throughout the talks today, stability data to ensure that your product is going to be stable over the course of your clinical study.
Thanks, Kristen. And this will go out to either one of you. One of you can jump in. But as you point out, it's controlled heterogeneity. But there, just let's pick a bispecific engager. I mean, there are a whole, all kind of different N and O link glycosylations, charge variants. There are a lot of different, modifications, post-translational modifications. How does a sponsor know what are the initial ones that I really need to address before I come into that IND? And I know you say, well, know your product and you'll know which ones, but do I really need to examine every potential, part of the heterogeneity as to whether that affects my potency assay? Is that the general expectation?
Hi, it's Wendy. Now you're going to get more knowledge as you go through. You can't know everything up front. So you look at the major components. You try to understand as best you can what they are. You know from the literature which features might be already understood as most critical. And then you try to keep your product as constant as you can while you refine it as you go forward. So you can't know everything up front, but you use the available knowledge to know what the most critical aspects are, and then you continue to learn as you go forward.
Great, thanks. And this is another question to you, Dr. Weinberg. It's kind of along the lines, and I'm going to paraphrase the question and let you kind of opine upon it. Pre-IND mechanisms of actions, changes to the IND mechanisms of actions, and when a Type C meeting would be more appropriate versus I'm kind of like annual reportable changes to me are very trivial things, and that if any substantive change wouldn't go into an annual reporting change. But there's kind of a question along those lines, and I wondered if you could kind of address that.
So the question is, what would come in as an annual reportable change versus an IND amendment or a meeting package type of thing?
Yeah, I kind of get the sense of that's the question.
So it's a broad question. Yeah, so the annual report, it has to do with a bar for safety and what has the greatest impact on safety and efficacy. For minor changes, those go into an annual report. If you're doing something like changing an assay, a major a manufacturing change that is more likely to have an impact on safety, purity, potency, and efficacy, then that would be an IND amendment. If it's you might want to come in and discuss it in advance, depending again on the risk. It's a risk assessment. It's risk based, and it really depends on the nature. We always say our products are case by case. It depends on the nature of the change. If somebody wants to come in and change a potency assay or broaden a specification, or I mean, those are a lot of the, change a column, those are not annual reportable items. Those are items that should be justified, and we would want to have a chance to look at and evaluate. And it might be something that would be useful to have a meeting for, to discuss in advance. Does that answer the question?
I think so. I hope so. It helps me. Kristen, this is directed to you. Can you just comment on the last year?
Did you?
Yeah, I heard you. No, you were great. It was good. Kristen, this is to you. The last year of COVID. Just give us a perception on your part.
Hi, did you hear my answer? I can't hear anyone.
Yeah, I'm going to go on. Can someone help Wendy on the other side, unless no one can hear me? This is to Kristen. Kristen, the last year of COVID, it's been busy. Have you guys managed to meet all the meeting requests that have been coming in?
Yes, so that's a very, I think it's a kind question. We have certainly seen an uptick in our workload with respect to the influx of our attention to COVID-related products, especially in our office, the Office of Biotechnology Products. However, with that said, I have not experienced any type of impediment on non-COVID related products receiving the attention that they would receive regardless of the current pandemic. We have just been trying to use alternate strategies within our office and our workflow to kind of manage the influx in those types of meeting requests. A lot of the meeting requests have been COVID specific. And so, that comes with the nature of the current landscape. But we've absolutely been having non-COVID product related meetings, and they have not been denied thus far.
Yeah, I try to help people understand that the work just keeps coming in, and we do it. So this is a question to Wendy, and hopefully she can hear me now. With regards to ADCs and antibody drugs conjugated to antibodies, whether that be ricin in the long past or one of these MMAFs or calichyamycins, are those reviewed substantively differently? Are they a similar review process?
Hi. So those are also BLA products. Those are collaborative reviews between the Office of Biotechnology Products, and the Office of New Drugs. So we work on those with ONDP, and so the antibody component, the part that gets that drug to where it needs to go, is really regulated the same way. We review it the same way. That's a critical part of its function, of its mechanism. And so we would expect the same kind of characterization for that pure product. And we work with the office, with our small molecule reviewer colleagues. They look at the small molecule. They look at the conjugation chemistry. We all look at it together, and we all look at the control strategies from our own perspective, right? So we would look at the antibody side, they look at the small molecule side, and the impurities related to each of those from our own perspective. There are additional aspects that would need to be considered for those kinds of conjugates. And so that would include looking at, a really critical aspect of it is the drug antibody ratio, and the stability of that linker, to ensure the safety. We also like to see, as part of characterization, how it's distributed, how the drug is distributed on the antibody, and that depends very much on the type of linker that's used. We also like to see from the antibody side not just a cell-based potency assay, but also a binding assay. Those can actually behave differently in terms of their stability indicating properties. So it's always nice to have those up front, to look at those parameters up front and follow it through development. So it's really a collaborative review process, but we have the same expectations. They're not lower standards for the antibody component because it's linked to a small molecule. There are still features of the antibody side that can still be contributing to the mechanism of efficacy. So it's the same, but collaborative with the small molecule reviewers.
Thanks, Wendy. I know it can get even more complicated when you throw in a nanoparticle and a liposome and a drug. But unfortunately, we have run out of time, which may be a good thing because the next question was about pentafunctional peptibody immune cell protagonists. Thanks, Wendy and Kristen, for some cogent enlightenment. It was greatly appreciated. And Lisa, I'm going to turn this over to you so you can introduce our next speaker.